3. Quick production use of automated techniques results in faster production of RABs. It usually takes about 8 weeks, whereas monoclonal antibody production by hybridoma technology requires 4 or more months. 4. Switching to isotype, the recombinant antibody fragment can be engineered into any isotype, such as zeta, IgM, and by combining proper constant domain. 5. Reproducibility, since the DNA sequence of the recombinant antibody is known, so batch-to-batch variation during manufacture is low. Unlike RABs, MABs generated by hybridoma technology may encounter variability during each production batch due to many factors, such as animal source, immortalized cells, accuracy of immunization, and so on. Antibody configuration. Antibodies consist of either monomeric or multimeric form of 4 chains containing 2 heavy chains and 2 light chains in a YY-shaped structure. Fv is a heterodimer of variable region of heavy and light chain, whereas Fab is a heterodimer of both variable and constant regions of heavy and light chains. Fc is a homodimer of constant regions of both heavy and light chains. The variable region is associated with antigen binding activity. Recombinant antibody technology uses smaller versions of antibodies to ease the display efficiency, rule out non-specific binding, and produce cost effectively. The antibody fragments used to generate libraries for the production of recombinant antibodies are domain antibodies, single chain fragment variables, and antibody binding fragments Fab. Domain antibodies consist of only the variable region of lighter heavy chain and are the smallest, 11 to 15 kDa, with 3 CDR in place of 6 from a full antibody structure. Due to smaller size, domain antibodies easily infiltrate tissues and are generally used in development of diagnostic kits. The scFvs are the combination of variable section of both heavy and light chain connected by glycine serine linker. The GS linker increases flexibility and stability and comprises of all the 6 CDRs as that of a full antibody. Due to their efficiency in phage display, scFvs are mostly antibody fragments used for the generation of antibody libraries. The scFvs are also used in diagnosis of prostate-specific membrane antigen, botulinum toxin, anthrax spores, and HIV-1. The Fab fragments 57 kDa are composed of entire fraction carboxyl terminus of the antibody. For phage display, fusion of Fab fragments, VHN. Figure 12.64 Production of recombinant antibody. Erythematosus and multiple sclerosis can be treated by RABs. Recombinant antibodies are found to be effective against antiviral diseases AIDS, hepatitis, Alzheimer's disease. RABs are able to cross the blood-brain barrier compared to MABs, and cardiovascular diseases. RABs with anticoagulant and antiplatelet functions. The application of recombinant antibody technology has a wide range of advantages over conventional monoclonal. The application of recombinant antibody technology has a wide range of advantages over conventional monoclonal antibodies. The most important is the avoidance of animal use for production. Recombinant antibody has a few limitations like expensive licensing, intellectual property rights, early investment in terms of time and energy, and production costs due to the use of high-throughput technology, but the hindrances are not invincible. Further development of recombinant antibody technology would accelerate the research in biomedical and biopharmaceutical. Vaccine. Vaccines are biological products that provide immunity against serious and deadly diseases. They are prepared from diluted or killed forms of microorganisms or their toxin or surface proteins. They induce the immune system of the body by recognizing the antigen and destroying it. They can be both prophylactic and therapeutic in nature, and the process of administering vaccines is known as vaccination, and the successful delivery of vaccines results into immunization. The term vaccine was coined by Louis Pasteur in 1846 in honor of Dr. Edward Jenner's work on cowpox virus as vaccine for smallpox. Vaccination begins right after birth with doses of hepatitis B viral antigen to protect the infant from hepatitis B up to the age of 65 or above. Pneumococcal vaccines are recommended for adults over 65 years old. Vaccination protects from microbial diseases, in a way has increased the lifespan. In this section, different types of vaccines will be discussed. Subunit vaccine. Subunit vaccines are the popular modern vaccine and are composed of specific components, purified macromolecules and A. The antibody genes are displayed in the phages. B. Canning, which involves incubation with specific antigen immobilized on a substratum, washing out of non-specific antibodies after incubation, elution of the specific antibodies. The eluted antibodies are introduced into bacteria to increase the volume of antibody. This step is repeated several times to enrich the antibody production. C. The antibody gene is inserted into the expression vector, transformed into E. coli and grown in an plate. The bacterial colonies expressing desired antibody gene were picked and grown. D. Antibodies produced were screened for specificity, which was expressed and purified. The recombinant antibodies were subjected to modification via error-prone PCR, DNA2FF link, site-directed mutagenesis, and exchange of CDR for increased affinity. Applications of recombinant antibody. RAB recombinant antibodies can be used in all areas where MABs are used, some of which are discussed below. Screening and detection. Recombinant antibodies conjugated with fluorescent tags can be used to detect concentration of several biological components, such as viruses, tumor cells, contaminants in food, and many others. Whole proteome analysis can be done using RABs. Diagnosis. The RABs can be used for diagnosis and monitoring cancer, also to find out vaccine effectiveness. A few reporter systems have been developed to enhance the sensitivity of ELISA-based assays viz. Antigen DNAzyme, immune RCA, and immune PCR. Another application of RABs in diagnostic is the development of lateral flow dipstick assay. This assay is usually found in the pregnancy test strip. Therapeutics. Recombinant antibodies are used widely in therapeutics. They can neutralize the bacterial toxins like botulinum toxin and staphylococcus enterotoxin, and snake venom. RABs can be used in liver cancer, lymphoma, leukemia, and multiple myeloma. Autoimmune diseases like rheumatoid arthritis. Systemic lupus delivery of subunit vaccine is mainly done in 2 ways. 1. Particulate delivery system. The main objective of this delivery system is to use a particulate carrier, similar to the pathogen, in order to deliver the antigen. Such resemblance induces the natural uptake and recognition, with a guarantee to transport the subunit vaccine to the antigen-presenting cells. Different sizes of particulate carriers ranging from nanometers to micrometer are used. The major advantage of employing the particulate delivery system is its potential to prevent the degradation of antigen from the harsh environment, thereby improving the antigen affinity. Other benefits of this delivery mechanism are that antigen and adjuvant can be delivered to both MHC class 1 and class 2 and also activate non-like receptor protein 3 NLRP3 inflammasome that boosts the immunity. Some of the commercially applied particulate-based vaccine delivery are emulsions including Freund's completed adjuvants FCA, Freund's incomplete adjuvants FIA, and Montanide for delivery of malaria and cancer vaccines. B. Alum was considered as gold delivery system earlier. Uses charged aluminum particles and includes several human vaccines. C. Virus-like particle VLP contains capsid protein or envelope. Commercially available VLP-based vaccines are Combavax HV for hepatitis B vaccines. Cervix GSK human papillomavirus vaccine and a few for cancer, HIV, and addiction therapies are under clinical trials. D. Bacterial dose consists of only cell envelope of gram-negative bacteria such as Vibrio cholerae and Escherichia coli. 2. Single-shot depot delivery system. This vaccine delivery system is based on sustained and continuous release of antigen and adjuvant for efficient prophylactic and therapeutic immune response. It is usually employed in 2 ways, such as hydrogel-based and implants. Hydrogels are the polymeric network, hydrophilic in nature. Sometimes even a non-toxic antigen derived from pathogenic microbes. These components are essential to induce a protective immune response and are considered as safe vaccines due to the lack of any live components. An efficient subunit vaccine comprises of a disease-specific antigen B, adjuvants, and C delivery system of the vaccine. Subunit vaccines are usually co-administered with an adjuvant such as toll-like receptor agonist to trigger efficient immune reaction. The adjuvant may activate the dendritic cells by binding to the pathogen recognition receptors PRR and stimulating inflammasomes. In general, the 3 forms of subunit vaccines are capsular polysaccharide, protein-based, and conjugate vaccine. 1. Capsular polysaccharide-based subunit vaccine. This vaccine targets the polysaccharide capsule layer around the microbe cell. This capsule acts as a protective layer for the pathogen, helping it evade the immune system of the host. Polysaccharide-based vaccine are less immunogenic, so requires a potent adjuvant for effective immune response. It is not much effective in children and induces interim immunity, no immune memory, e.g., vaccine for meningococcal diseases, pneumococcal disease. 2. Protein-based subunit vaccine. A specific isolated protein of the microorganism is used as a vaccine, and this vaccine presents the antigen without the viral particles. It may undergo denaturation resulting in non-specific binding, e.g., a cellular pertussis vaccine app, which contains an activated pertussis toxin. Hepatitis B, Hep B contains surface antigen of hepatitis B virus. 3. Conjugate subunit vaccine. It is generated by linking the capsule polysaccharide of the bacteria and a carrier protein. The carrier protein can improve and modulate the immune response of the weak polysaccharide vaccines, e.g., Haemophilus influenza type B conjugate, Hib pneumococcal conjugate. Vaccines, and hence are less immunogenic and require adjuvants or carrier proteins for a strong immune response. Methods for designing the protein epitope. There are various points to be considered while designing an epitope. The first step is to identify an epitope that activates the immune system and exhibits long-lasting immunity. The epitope chosen may not be immunodominant all the time. For example, though nectar Americanus APR1 protein does not induce immunity against hookworm and produce neutralizing antibodies, it was recommended as a potential peptide vaccine candidate. Other features to consider while designing peptides are hypersensitivity responses of the epitope and the inclusion of the conserved amino acid sequences. The length of the B-cell epitope may vary, and longer epitopes are essential to maintain the native conformation, for which the epitopes are flanked at both the C and N terminals to induce a secondary structure. An alternative to flanking is stapling of the short peptide to induce desired conformation. Here the peptide is stapled by incorporating chemical bonds between the amino acid side chains. Cyclization is also one of the strategies to control the 3D structure and conformation. HIV vaccine development follows cyclization of the peptide. These three strategies can be followed to maintain the conformation and to prevent the epitope from enzymatic degradation. A few modifications can also be done for improvement of the stability in in vivo conditions, such as acetylation of N-termini, amidation of C-termine, or modification of sequences by incorporating a natural amino acid L-cyclohexylalanine, aminocaproic acid, and dialanine, or conjugating with lipid carbohydrate at both the can N-terminus, figure 12.65. Delivery of peptide vaccine is similar to that of subunit vaccines, yet particulate vaccine delivery system is mostly employed. Peptide vaccines are yet not available in the market and several under clinical trials, approximately 306 peptide vaccines, mostly cancer vaccines. They are quite a new type of vaccine in the world. And are apt for drug, vaccine delivery, and tissue engineering. The specific features that make hydrogel a delivery system are easy administration, easy incorporation of vaccine, and its soft tissue-like and sustained releasing properties. A few examples of hydrogel-based vaccine delivery are triple copolymer, polyethylene, polycaprolactone, and polypropylene oxide for basic fibroblast growth factor (BFGF) delivery, beach-based hydrogel biscovyl TM for delivery of Haemophilus influenzae type B vaccine, and C-peg-based hydrogel for hepatitis B vaccine delivery. Implants are commonly used for drug delivery but are recently considered even for sustained release of vaccines. Polymer or lipid implants are being used for subunit vaccine delivery. The development of effective, safe and marketable subunit vaccine depends on the immunogenicity of the antigen, the adjuvant, the delivery system employed, and the route of administration. The commercial vaccines available are generally administered subcutaneous, like whole cell subunit vaccine, intravenous cancer vaccine, intradermal influenza vaccine, oral whole cell vaccine, nasal, and pulmonary influenza vaccine routes. Subcutaneous route is the least painful and a large dose of vaccine can be administered. Many of the subunit vaccines are in a clinical trial phase, and more research is required for the development of successful and efficient subunit vaccines to prevent and treat deadly diseases. Peptide vaccine. Peptide vaccines comprise of only specific protein sequence, usually 20 to 30 amino acids, that have the potential to induce both humoral and cell-mediated immune response. The use of such a specific epitope reduces the probability of allergenic or reactogenic responses. The peptide vaccines can be used as broad-spectrum vaccines effective for different serological variants of disease pathogens. This can be obtained by using the conserved protein sequence between the serobars as an epitope. Peptide vaccines are a type of subunit of vaccination and require lots of research for better understanding of their immunogenicity, selection, and proper designing of epitopes to avoid reactogenicity. Peptide vaccines demonstrate to be promising immunotherapy triggering cell-mediated immune response and effective prophylactic vaccine inducing humoral immunity. Figure 12.65 methods of designing epitope. A. Stabilize conformation and B. Improvement of its stability in vivo by addition of unnatural amino acids or carbohydrate, lipid moiety, or introducing amino acetyl group. Live attenuated vaccines. The first vaccine successfully developed was smallpox vaccine in 1796 by Dr. Edward Jenner, which is a live vaccine with attenuated Virginia. Live attenuated vaccines labs contain the weakened form of the live pathogen which causes the disease. This type of vaccine produces robust and long-term immunity, but since includes live viruses, though weakened may not do well in the immunocompromised people. Despite the fact that live attenuated vaccines have been very successful, the safety and efficiency of these vaccines is dubious. Hence, the root and period of immunization of live attenuated vaccines are important for their utmost efficacy. For example, mumps and polio vaccines are administered in mass campaigns to prevent dissemination of the virus and reduce the affected population. The major challenge in developing live attenuated vaccines is the choice of a vaccine strain with right features. Live attenuated vaccines are developed on the basis of observations made from clinical experiments. The mechanism for the development of labs is either chemical mutagenesis or laboratory experiments or recently, genome analysis. The method of attenuation may result in problematic quality control and reversion into virulence. Labs elicit T-cell dependent response by presenting the attenuated live virus bacteria to MHC class 1 and MHC class 2 generating CD8 plus and CD4 plus T cells, respectively. Live vaccines are observed to produce strong CD8 plus cytotoxic T cells. CD4 plus T cells induced by the live vaccines result in macrophage activation and control, reduce pathogens such as mycobacterium tuberculosis and varicella zoster. The commercially available immunity by both the antigen and the plasmid DNA itself. Following the incorporation of plasmid DNA into host cell, the mammalian promoter activates the transcription of the specific antigen gene. The expressed antigen or process in the peptide binds to MHC class 1 and MHC class 2 and are presented on antigen-presenting cells, such as dendritic cells DCs, to stimulate T cells. The specific antigen can activate B cells for antibody generation. DNA vaccine may be injected into the stromal cells or directly delivered. When transfected into muscle cells, the antigen are captured by DCs and then cross-presented to CD8 plus cytotoxic T cells. While if directly delivered, all the steps like the expression, processing, and secretion of the antigen are carried out by DCs presented to CD8 plus CD4 plus T cells. In case the expressed antigen is captured by B cells with the help of helper T cells, antigen-specific B cells are generated. The dsDNA incorporated into the cytosol induces immune response via key players like TBK1, IKK epsilon, and STING. The DNA plasmid along with cDGMP, cGAS, and RNA polymerase III binds to cytosolic sensors and triggers the production of type 1 interferons IFN via STING TBK1 pathway where STING induces TBK1 activity, phosphorylating IRF3 or STAT6, and dimerized IRF3 STAT6 penetrate into the nucleus thereby activating the expression of IFN. STING stands for stimulator of interferon genes, detects the nucleic acid from intracellular pathogen when cells are infected by a pathogen. DNA vaccines can be delivered in many ways, such as mucosal injection, electroporation, gene gun, and pressure injectors. Most of the pathogen enters via mucosa, so mucosal injection offers an advantage to administer the vaccine, increasing the potency of the vaccine. Electroporation is a commonly employed delivery system of DNA vaccine where a small electric current is applied in vivo, because it increases the uptake of the plasmid DNA into the cells resulting in both humoral and cell-mediated immune responses. To avoid DNA degradation and enhance uptake, DNA can be encapsulated into microparticles. Adjuvants, such as labs recommended by WHO or BCG, oral polio vaccine, measles, rotavirus vaccine, and yellow fever vaccine. BCG is a live vaccine based on attenuated pathogenic bacteria, Mycobacterium bovis. Some of the concerns related to live attenuated vaccines are: though reversion of pathogenicity of the attenuated pathogens is rare, if this happens, it may lead to disease progression worse than before. Additionally, immunocompromised individuals, such as HIV-infected patients, may not react to labs. Therefore, live vaccines for chronic diseases like HIV and HCV are not developed. Live attenuated vaccination and prolonged infection may lead to local lymphadenitis or transmission of the infection. Labs cannot be administered to pregnant women in order to avoid fetal damage. Most of the labs need a cold chain supply to maintain their activity, so the immunization program may be a failure where there is no cold storage facility. Some labs are lyophilized. Reconstitution of such labs may reduce their activity if not carried out as required. Nevertheless, live attenuated vaccines are the cheapest vaccine presently available and have been successful in eradicating smallpox. Poliomyelitis is also close to eradication. Administration of lab for measles has controlled the disease in most parts of the developing countries, and in the Western Hemisphere, rotavirus vaccines have reduced mortality due to rotavirus. However, a better understanding of the history of live attenuated vaccine would provide pieces of evidence that would further help in developing safe and efficient live attenuated vaccines. DNA vaccine. DNA vaccines were introduced almost 2 decades ago with the hope to modulate the immune response against chronic diseases like cancer, HIV, hepatitis B, and malaria. A DNA vaccine consists of plasmid DNA that code for a specific antigen under a promoter activating the expression. DNA vaccine may stimulate aluminium salts and genes encoding for cytokines, and co-stimulatory molecules added to the plasmid backbone are shown to boost potency of a DNA vaccine. In a preclinical setting, it was observed that if DNA vaccine is combined with a vector system like adenovirus, the protective efficacy of the vaccine significantly amplifies. Figure 12.66 The mechanism of action of DNA vaccines. Component I. DNA plasmid transfection into the cytosol, i.e., by electroporation elicit both humoral and cellular immune responses, antigens expressed in muscle cells and DCs. These antigens are processed and presented either by direct or cross-presentation, thereby stimulating CD4 or CD8 T cells to generate cytotoxic T cells and T helper cells, respectively. The antigen can directly induce B cells producing antibodies. Component 2. The transfected DNA plasmid and its metabolites such as cGAS, cDGMP, RNA polymerase III, and uric acid function in a network with DNA receptor present in the cytoplasm and trigger the STING TBK1 IRF pathway to release of type 1 IFNs. Several clinical trials for vaccine development against HIV, cancer, influenza, and hepatitis B have been done but resulted in unsuccessful outcomes. DNA vaccines experience low immunogenicity but are safe. So if the immunogenicity is increased, it is believed to develop successful DNA vaccines, especially for HIV and HCV in the future. Licensed DNA vaccines such as West Nile viruses in horses and canine melanoma for veterinary application is available since 2005. Understanding the working mechanism of DNA vaccines and selecting highly immunogenic antigen may result in successful DNA vaccines. Vector vaccine. Vaccines that carry live viruses or bacteria which transport desired antigen of the pathogen to the human cells for effective host immunity are characterized as vector bacteria vectors. The heterologous antigen can be a bacterial protein or inserted into the plasmid under the control of viral promoter. When intracellular bacteria are used, the expressed and processed antigen is delivered to MHC class I producing cytotoxic T cells. Salmonella, Shigella, Mycobacterium, and Listeria are used in vector vaccines. 3. DNA and RNA vector DNA was measured as insufficient to induce immunity, as because it may undergo degradation if directly transfected into the cell. But the successful event of plasmid DNA vaccine proved it wrong and it was reconsidered to be used as a potent vector vaccine. RNA has been used in the development of vaccine and immunotherapies, but it is prone to degradation as compared to DNA, so it is required to stabilize RNA encapsulation or formulations. The potency of DNA vector vaccine can be enhanced by codon optimization, inclusion of CpG motifs, change of promoter for amplification in gene expression, and addition of co-stimulatory molecules, cytokines. To increase the potency of the vector vaccine, prime boost protocol can be followed. In prime boost, an initial prime dose of one type of vaccine, such as DNA vaccine, expressing a particular antigen is administered, followed by a boost with introducing vector vaccine expressing the same antigen expressed by prime. Preclinical and clinical studies show extensive use of prime boost modality of vaccination. Phase 2 HIV vaccine trials include a prime of DNA plasmid and a boost of adenovirus vector. The vector-based vaccines licensed till now are applicable for animal diseases, such as MVA vector rabies vaccine, vector-based DNA vaccine, such as hematopoietic necrosis virus in fish, West Nile virus horses, and melanoma in dogs. The vector vaccines can activate both prophylactic and therapeutic response. Additionally, the characteristics of a vector vaccine make it a strong form of vaccination. Vaccines. These types of vaccines have the potential of delivering different epitopes into the antigen-presenting cells to bind to the MHC class I, eliciting cytolytic immune response. Though vector vaccines generate cellular response, that does not mean it is insufficient to induce humoral immune response. Various transfection techniques is mentioned in the case of DNA vaccine and vectors such as viral vectors, bacterial vectors, and nucleic acid vectors. 1. Viral vectors. Several viral vector vaccines have been developed, of which many are in clinical trials. The characteristics to be considered while selecting a particular virus as vector include the efficacy of expression of different epitopes, persistence of the virus so that it can induce long-term immunity in the antigen-presenting cells to which the transfected vector virus will be delivered. Pox virus is employed as one of the potential vectors because much is known about the safety and efficacy of smallpox vaccine. Also, its ability to carry heterologous genes for the desired antigen requires a brief period of time for antigen production, low integration risk, and wide response in mammalian cells. The first licensed vector vaccine was for veterinary application, in which rabies virus was incorporated into smallpox virus vector. HIV vaccine is one of the pox virus vectors under clinical trial. Adenovirus vectors are less effective as adenovirus antibodies Ad5 already present in the individual can bind to the vector, reducing the vector's activity. For example, individuals turned out to be more susceptible to HIV infection if they had high titers of Ad5 antibodies. Hence, reusing adenovirus requires the removal of certain adenoviral genes in order to avoid undesired binding and increase the potency of adenovirus vector vaccine. 2. Bacterial vector. Attenuated forms of bacteria are used as vector. Bacteria can be easily administered orally, which make them a suitable vector. In the different food industry sections which undergo food processing are baking industry. During the dough phase, i.e., when flour, yeast, and water are added to prepare the dough, enzymes, especially amylases, are added externally to improve the taste, texture, and perishability. In baking industry, starch is the predominant component present in all baked goods. Alpha amylase hydrolyzes starch into short chain dextrins, while beta amylase breaks down short chain dextrins into disaccharides like maltose, which is fermentable. Fungal amylase and bacterial amylase are used for two different purposes. Fungal amylase acts upon degraded starch, while bacterial amylase catalyzes the latinized, heated starch and water mix. Other enzymes used in baking industry are lipoxygenase, which reduces the yellow color of the flour resulting into white bread and also improves loaf size. Xylanase, protease, and lipase are also used to ease the handling and manufacturing process, makes the bread softer, have an anti-scaling effect. Alcohol industry. Malting and beer industry requires supply of exogenous enzymes that can hydrolyze starch, proteins, pectin, cellulose, and hemicelluloses. Commonly used enzymes in alcohol production are polylanase, amylase, amyloglucosidase, and glucanase. Clarification of beer is done by the proteolytic activity of protease, papain, and lactase. Dairy industry. Aspartic protease is important for cheese production, where it is obtained conventionally from the stomach of young calves in form of a mixture of chymosin and pepsin. Recombinant chymosin is expressed by microbes for faster and maximum manufacture. Chymosin and pepsin coagulate milk and ripen cheese. Proteolytic activity. Plant-based aspartic proteases, cardosins, cyprosins, and clot milk. Lactases are used in processing dairy products, thereby reducing lactose intolerance. Enzymes. Enzymes are biocatalysts that accelerate chemical, biological, and metabolic reactions. Enzymes are mainly proteins or polypeptides, except the RNA enzymes also called ribozymes. Enzymes are substrate specific and convert substrate into product. Enzymes are the most used biological molecules in various fields such as agricultural industry, pharmaceutical, food industry, and so on. Some of which are explained in this section. Enzymes in food and beverage industry. Enzymes have been used as a tool in food industries for centuries. Enzymes are extracted from animals, plants, and microorganisms for food processing. Enzymes from plant sources used in food and beverage industry include bromelain, papain, alpha amylase, beta amylase, ficin, lipoxygenase, beta glucanase, and actinidin. The enzymes of animal origin are catalase, rennet, and lipase. Enzymes from animal sources are complex and expensive and may have the potential to cause disease while handling and production. The enzymes from microbial sources are widely used and mostly preferred because the production of microbial enzymes is cheaper, faster, and large scale. The microbial enzymes are safer and more stable than the plant and animal enzymes. Also, the microbial-based enzymes possess generally regarded as safe broth label. After recovery of the enzymes from the culture broth, the enzymes are added to the food preparations and perform the required functions like upgrading the flavor, digestibility, and nutritional value. In food and beverage industry, the enzymes are either partly purified or bulk enzymes, so processing of complex food requires care to avoid any unwanted enzymatic activity. The enzymes to be used in food industry must possess the capacity to withstand a wide range of environmental conditions such as temperature per picohenry. For example, protease used for meat tenderization must be active at both room temperature and cold storage conditions, but inactive at cooking temperature in order to avoid over-tenderization. 3. Catalase. It helps in decomposition of hydrogen peroxide. It is used as a preservative in milk processing to avoid oxidation and increase the shelf life of food. Catalase is produced by Aspergillus niger, Micrococcus luteus, and from the liver of cow. 4. Proteases hydrolyze the peptide bonds present in proteins and polypeptides. Plant proteases include bromelain, ficin, and papain, which are widely used in coagulation of milk, meat tenderization, and alcohol production in food and beverage industry. Proteases also enhance the flavor, nutritional value, and digestibility of food. Microbial acidic proteases are used in brewing and increase digestibility of wheat gluten. 5. Esterase. It breaks esters into acid and alcohol and also hydrolyzes short-chain acylacerols. It is used in both food and beverage industries. It induces flavor and fragrance into the food and modifies oils and fats in fruit juices. Enzymes for agriculture, biomass, and biofuel. Enzymes for agriculture. Agricultural yield primarily depends on the soil enzymes. Plants need essential macronutrients C, H, O, N, and P, secondary nutrients Ca, Mg, and S, and micronutrients Cu, Fe, Mn, Mo, and Zn for healthy growth, and your soil enzymes play an important role by regulating the biogeochemical cycle of the macro and micronutrients. Rhizosphere is the primary microbe region where most of the crucial enzymatic activity is executed. The physical properties of the soil and the microbial diversity in the soil influence the crop yield. Sandy soils are porous and are home to low-density microbes compared to the clay soils. Soil microbes secrete enzymes required for healthy plant and higher crop yield. Sandy soils are supplemented with manure and biofertilizers that finally improve microbial density and increase the nutrient content. Fruit and vegetable juice industry. Pectinase digests the pectin present in fruits and vegetables, which further breaks the jelly mass formed in the juice, decreases the viscosity, and thus clarifies the juice. The bittering of citrus juice is done by marinase, which is a complex of alpha rhamnosidase and beta glycosidase. Meat industry. Bromelain, ficin, and papain are used to tenderize meat. However, these enzymes are thermostable, i.e., do not denature even during cooking. Microbial-based enzymes, subtilized in a neutral protease, have an advantage in that they are active at low temperatures and denature upon cooking. Transglutaminase enhances the texture, keeps the meat intact, and increases the shelf life. As discussed above, the application of enzymes in food processing in different industries. Here are some of the commonly used enzymes applied in food and beverage industry elucidated below. 1. Alpha amylase. Alpha amylase is found in all living systems which digest starch by hydrolyzing the alpha-1 4 glycosidic bonds resulting in short-chain dextrins. It requires calcium ions for its activity, stability, and integrity. In baking industry, alpha amylase is used as a flavor enhancer and it improves the bread quality. In starch industry, starch undergoes enzymatic conversion by alpha amylase to produce glucose and fructose syrup. Alpha amylase is also used in ethanol production and clarification of fruit juices. 2. Lipase. It hydrolyzes lipids leading to release of free fatty acids, glycerol, and monodiacylglycerols. It is found in the stomach and pancreas of humans and other animals, while microbes, such as yeast, bacteria, and fungi. Lipases are used as flavor enhancers in butter, margarine, and cheese in dairy industry, while in brewing industry, lipases alter the aroma. Immobilized lipases are employed as a biosensor to detect pesticides, triglycerides, and blood cholesterol. Linocellulosic biomass is recalcitrant in nature and difficult to degrade, so conversion to ethanol is a challenge. Bioconversion has an upper hand over the mechanical and chemical methods of disintegrating linoselluloses. Enzymes, such as cellulase, xylanase and glucose-tolerant enzyme, and beta-glucosidase are mainly used for hydrolysis of linoselluloses. 1. Cellulase hydrolyzes cellulose and comprises of three enzymes of glycosyl families, such as endoglucanases hydrolyze internal glucosidic linkage cell biohydrolases, and beta-glucosidase, where endoglucanase and cell biohydrolase perform primary hydrolysis while beta-glucosidase performs secondary hydrolysis. These three enzymes function synergistically, fungi are efficient in the production of cellulytic enzymes. 2. Xylanase hydrolyzes xylane, a component of hemicelluloses. Unlike cellulase, xylanase can easily act upon the xylane due to its simple crystalline structure. Xylanase are produced by fungi, such as Trichoderma rheaceae, Humicola insolens, and Bacillus spp, which are active from 40C to 60C similar to cellulose. Xylanases also require several enzymes for complete hydrolysis of xylane, which includes endo-1, 4-beta-xylanase, alpha-arginine esterosis, and beta-xylosidase. These enzymes act upon xylane in a sequential manner. 3. Lactase and peroxidase act upon the lignin by catalyzing its degradation. 4. Glycoside hydrolases are the important enzymes which hydrolyze biomass and are produced by a variety of microbes, such as archaea, bacteria, fungi, protists, plants, and animals. These enzymes act upon the main chain of cellulose and hemicelluloses and are of industrial importance. To make the glycoside hydrolases induced bioconversion cost-effective, improvement of properties, higher stability at extreme condition of temperature and pH and increased specificity, and a cocktail of the enzymes is used. Bacteria like Cellulomonas, Clostridium, Bacillus, Dermonospora, Ruminococcus, Bacterios, Erwinia, and others produce glycoside hydrolases which, for raising agricultural products, in addition to soil properties, enhancing photosynthetic efficiency also increases crop yield. The important soil enzyme complexes include the enzyme classes, such as oxidoreductases, lyases, and hydrolases. A few of the enzymes are discussed below. 1. As mentioned in the previous section, amylases break starch into glucose units. They play an important role in carbon cycling, making carbon available for plant uptake. 2. Cellulase digests the cellulose which is abundant in nature, releasing glucose as a final monosaccharide. 3. Phosphatases are important in plant signal transduction pathways, such as stress response, cell differentiation and metabolism, and act upon the peak chemical fertilizer. Phosphatase hydrolyzes the phosphate bonds releasing orthophosphates. Fungal phosphatases are alkaline in nature while bacterial phosphatases are acidic. 4. Protease hydrolyzes peptide bonds finally resulting into amino acids which are the nitrogen source. Thus protease assists in nitrogen assimilation which is important for crop production. 5. Rubisco is an important enzyme in photosynthesis and photorespiration regulating CO2-O2 ratio. When the CO2 concentration rises, photosynthesis and growth rate increase, thus affecting the agricultural productivity. Genetic engineered Rubisco, which increases photosynthetic efficiency, was included in a wheat variety in soybean and observed to produce increased yield. Biomass and biofuels. Lignin is a common organic component of linoselluloses of the plant cell wall and waste from food industry. The linoselluloses are the most abundant biomass on the earth and are enzymatically hydrolyzed to produce biofuels. Figure 12.67. Linosellulose is a carbohydrate complex consisting of cellulose, hemicellulose is tightly bound to lignin. Cellulose and hemicellulose upon enzymatic action are broken down into monomeric sugar units, which produce bioethanol by microbial fermentation. Lipase and also whole cell catalyst. The use of whole cell catalyst reduces the cost as this does not require extraction and purification steps. Lipases are immobilized on supports, such as decotyl sapobeds, chitosan beads, periodic mesoporous organosilica, mesoporous carbon beads, green coconut fiber, and activated agarose gel. There are other methods of immobilization, which include cross-linking enzyme aggregates, magnetic particles carrier, and protein-coated microcrystals. Immobilized lipases are usually used for large-scale industrial production. Second-generation biofuels are derived by hydrolysis of biomass. The enzymatic hydrolysis of biomass is discussed above. Algae store large amounts of oils in the form of triacylglycerols during nitrogen stress. The triacylglycerols are then converted to biofuel. In a recent study, it was observed that glycerol 3-phosphate acyltransferase family, GPAP abundant in the aldocyanidioxide primarily, increases the production of triacylglycerol by almost 56 times. This rise in triacylglycerols results in high yields of biofuels. The different types of biofuel produced by first, second, third, and fourth generation processes are: a biogas. It is a mixture of gases upon breakdown of organic matter. Enzymatic production of biogas production includes mainly the use of subtilizin, protease, and cellulase. Beet syngas, a mixture of CO, H2, and other hydrocarbons obtained from biomass. Astagens are specifically used for fermentation of syngas and use the acetyl-CoA pathway, Wood-Lindahl pathway, for acetic acid and ethanol production. C. biodiesel. It is a common biofuel and is produced via transesterification of biomass. D. Other biofuels include vegetable oil, bioethers, and green diesel, similar properties to that of petroleum-based diesel, bioethanol, and biomethanol. Are commercially available. Trichoderma rheaceae is one potent commercial glycoside hydrolase producer among fungi. The utilization of enzymes in biomass hydrolysis is a costly process, so there are a few strategies which can be adopted to make the process cost-effective. The use of genetically engineered microorganisms with elimination genes encoding for unwanted enzymes and specific alteration in the basis of desired genes to improve specificity and stability is one of the strategies to reduce the cost. Another strategy is to introduce mutagenesis via DNA HUFF link, XN2FF link, error-prone PCR in the target gene to enhance the catalytic activity of the enzymes. Pure culture used for biomass hydrolysis is costly and a particular strain may not be efficient in the production of high titer of the enzyme, so culturing two or more compatible strains or cocktails may result in higher yield. Biomass hydrolysis using enzymes is a potential and environment-friendly approach toward biofuel production. As the term suggests, biofuels are the fuel obtained derived from biological sources, i.e., biomass. The main sources of biofuel are plant, agricultural, industrial, and domestic wastes. Reports suggest that in 2018, biofuels contributed 3% of the world's total fuel for road transport. There are 4 generations of biofuels, namely a first generation includes biofuels produced by transesterification of crops dedicated for biofuel production. B. Second generation derived from linosellulosic biomass. C. Third generation from algae, microalgae. D. Fourth generation similar to third generation but only differs in that they do not require biomass destruction. The first generation of biofuel can be produced by non-catalyzed reaction or chemical catalyzed reaction and enzyme catalyzed reaction. Lipase is used in enzyme catalyzed reaction for biofuel production. It is produced by bacteria, fungi, and yeast. Free lipase. Immobilized. Figure 12.67 schematic representation of conversion of biomass into biofuel. The linosellulosic biomass undergo processing and pretreatment prior to the important downstream processes. The downstream processes include enzymatic hydrolysis of the pretreated biomass, fermentation (SHF), separate hydrolysis and fermentation (SSF), simultaneous saccharification and fermentation (CPP), consolidated bioprocessing (DMC), direct microbial conversion. The final product obtained are recovered from the bioreactor and purified. Enzymes for antibody research. Antibodies have been accepted as therapeutic tools for the treatment of various diseases. Still, researches on antibodies are in progress for modification to increase their efficiency. Enzymes such as proteases, glycosidase, and transaminase are used for antibody research. Antibodies, when conjugated with drugs, are found to be effective against diseases like cancer, and this concept has gained attention after the antibody drug conjugates ADC, Kadcyla, and Adcetris became available. ADCs proved to be a promising therapeutic tool and better than the antibody therapy and conventional drug treatment. In this section, we would specifically focus on enzymatic development of antibody drug conjugates as part of antibody research. The ideal drug to antibody DAR ratio for most ADC is 2 to 4 but may vary depending on the features of the drug. In ADCs, the antibody fragments and their mimetics are generated from the antigen binding fragment Fab, single chain variable fragment SCFE, and single domain antibody or nanobody VHH. The nanobodies are derived from camelid heavy chain which inhibits antigen binding, delaying malignancy. Other antibody mimetics include ankyrin repeat proteins, leucine-rich repeats for cavities, and Staphylococcus protein A phagoides, which easily penetrate into the tumor cells due to smaller size. A few enzyme-mediated modifications of antibody mimetics are needed to overcome its short half-life and inability to pose antibody-dependent cell cytotoxicity (ADCC). Formal glycine enzymes (FGEs) are produced by both prokaryotes and eukaryotes, which introduce formal glycine replacing cysteine and add an aldehyde group to the ADCs. FGE uses sulfate as substrate so attack the cysteine residue. Genetically engineered microbes are used to produce HFGEs. This enzymatic reaction, in fact, tags the ADCs with an aldehyde group enhancing antibody drug activity. Sortase is expressed by gram-positive bacteria, where it recognizes LPXTG X equals glutamic acid, aspartic acid, lysine as the presence of ZN2+ and 3 amino acids long tag is ligated at the C terminus. Engineered enzyme like spilidase derived from Streptococcus pyogenes is also used to catalyze tagging of antibody. Spycatcher consists of 3 types of antibody tagging enzymes, such as SpyTag, KTag, and spilidase where spilidase ligates the 2 tags generating ADC. Other than enzyme-based antibody labeling of antibody drug conjugates, enzymes are also employed for different antibody research. Immunoglobulin proteases, IgM proteases, are used to generate Fab and Fc fragments which can be engineered to obtain desired antibody fragments to be used for therapeutic purposes. Immunoglobulin proteases, IgM proteases, are used to generate Fab and Fc fragments which can be engineered to obtain desired antibody fragments to be used for therapeutic purpose. Arginine-specific protease can be used for antibody characterization. Glycosidase can be used to hydrolyze the end glycans of Fc region of making it more useful therapeutic protein. Substrate motif. It hydrolyzes the peptide bond between threonine and glycine, thereby adding a nucleophilic tag of 2 to 3 glycine residues to facilitate peptide linkages. Transglutaminase transfer the acyl group to the side chain of a glutamine residue in presence of CA2+. It is produced by both eukaryotes and prokaryotes, where mammalian transglutaminase calcium-dependent enzyme wild transglutaminase from Streptomyces mobarensis is neat calcium ion. Alpha-tubulin is catalyzed by tubulin tyrosine ligase to add a tyrosine residue at C-terminus, which increases the solubility of the protein. This ligation process is also known as TUB-TAG. Such TUB-TAG antibody drug conjugates are advantageous in terms of their polarity. Proteases like tripsilidase and subtilidase induces peptide formation along with hydrolysis. Tripsilidase catalyst is in Figure 12.68 enzyme-based antibody labeling of antibody drug conjugates. Information in the living cells. These signals are amplified via feedback mechanism, binding of the signaling molecules and the receptors, and interconnection between the receptor molecules. Another mechanism is the push-pull network, which is found in both prokaryotes and eukaryotes. Here, two enzymes covalently bind and modify a component in the opposite manner. For example, kinesis phosphorylate a component of a pathway while phosphatase dephosphorylate the same component. A few important enzymes for cell processes are discussed below. 1. Acetylcholinesterase 8 hydrolyzes choline esters in the body and is abundant in neuromuscular joints. This enzyme has a robust catalytic potential where one molecule of acetylcholinesterase can break down acetylcholine at the rate of 25,000 sec. During neurotransmission, the presynaptic neuron releases 8 into the synaptic cleft, which then binds to the receptors on the postsynaptic cleft. Inhibition of acetylcholinesterase results in acetylcholine buildup at muscarinic receptors of smooth muscle, salivary glands, exocrine pancreas, and intestine. Irreversible AKE inhibitors, which include insecticides, nerve gases, etc., may cause muscular paralysis, bronchial constriction, convulsions, and also death by asphyxiation. Reversible AKE inhibitors are used to treat certain diseases like Alzheimer's disease, glaucoma, myasthenia gravis, and nervous system disorder. The common reversible AKE inhibitors used for treatment are tacrine and donepezil. 2. Carbonic anhydrase catalysis. The interconversion between carbonic acid and CO2 and H2O. This enzyme is crucial for cell processes as it aids in regulating the pH and fluid balance and CO2 transportation. Carbonic anhydrase plays organ-specific roles such as in stomach carbonic anhydrase produces acid lining, while in the kidneys and eyes it regulates the water content. In lungs it controls the exhalation. It is one of the fastest enzymes with a catalytic rate ranging from 10.4 to 10.6 reactions sec. There. Antibody modification by A-formal glycine generating enzyme, beat sortase D-tubulin tyrosine ligase E-tripsilidase F spilidase. The details of medication methods are discussed in the text. Enzymes for cell biology. The entire cellular processes inside the body are catalyzed by enzymes. Enzymes allow the cell to perform the metabolism, neurotransmission, signaling, apoptosis, and miscellaneous functions which are crucial for the living system. The synthesis of enzymes must be regulated as the requirement of cells may differ. For example, a liver cell does not need a muscle enzyme. Some enzymes are only synthesized when required. Some are produced by particular cells, while some are found in all the cells. In metabolic pathways, the product of one enzyme is used as a substrate of another enzyme. In absence of enzymes, metabolism does not proceed through the same steps and meet the cell needs. Enzyme activity can be controlled by regulation, post-translational modification, quantity of enzymes, compartmentalization, and organ specialization. Negative feedback mechanism is a regulatory mechanism of an enzyme where the concentration of the end product is regulated by its own concentration. For example, the production of blood glucose level is regulated through a negative feedback mechanism. When the glucose level is high, insulin stimulates the enzymes involved in glycogen synthesize, while when the glucose level is low, glucagon induces glycogenolysis. Post-translational modification like phosphorylation of glycogen synthase aids the control of synthesis and degradation of glycogen, enabling the cell to sense the change in blood sugar level. Neurotransmission is a process where presynaptic neurons produce neurotransmitters to bind to the receptors of postsynaptic neurons and thereby activate the nerve impulses. Acetylcholinesterase breaks down the neurotransmitter acetylcholine into acetate and choline esters, thereby triggering the postsynaptic nerves, muscles, and exocrine glands. Signal transduction is an important mechanism for transferring signals. Diseases caused due to deficiency of specific enzymes include Gaucher's disease, defective glucocerebrosidase, pyruvate kinase deficiency, and congenital adrenal hyperplasia deficiency of 21 hydroxylase, 3 beta hydroxysteroid dehydrogenase, 11 beta hydroxylase, and cytochrome P450 oxidoreductase, glucose 6-phosphate dehydrogenase deficiency. Enzymes for chemical processing. Enzymes are used in various fields due to their specificity and biodegradability. But in chemical production, it was found that only a few natural enzymes are used and engineered enzymes are preferred. It is because of the fact that Baumar has pointed out that most of the enzymes have suboptimal properties for processing conditions. Literature cites that a large number of enzymes can be used for chemical processing, but only a few, about 1% of the total enzymes, are practically used for industrial production. Currently, there are various methods for creating novel enzymes and improving the catalysis power. Gene encoding for the desired enzyme is overexpressed in the microorganism by using optimized gene and vector system for high enzyme yield. In addition to the genetic engineering of the microorganism, improvement of the efficiency of enzyme can be done by protein engineering, which increases the reaction rate, substrate specificity, and stability of the enzyme. Bioprospecting is one of the methods where unculturable microbes are used by employing metagenomic techniques. Metagenomic library offers the detection of enzyme activity. Metagenomics for enzyme utilization in chemical production includes monitoring environmental samples and insertion of both small size and large size clonal libraries to activate the transcription of enzyme coding gene of the biosynthetic gene cluster. Another approach to enzyme application in chemical processes is directed evolution of enzymes, which includes the construction of mutant libraries, screening, selection, and isolation of the gene with desired mutations. The directed evolution of alcohol dehydrogenase and glucose dehydrogenase led to the production are 5 distinct carbonic anhydrase families that include alpha, beta, gamma, delta, and zeta. Alpha type is found in humans. Deficiency of carbonic anhydrase leads to autosomal recessive disorders like osteopetrosis, cerebral calcification, and renal tubular acidosis. Acetazolamide, dorzolamide, and methazolamide are a few carbonic anhydrase inhibitors which are chiefly used to treat glaucoma. Also can be employed as antidiuretic, antiepileptic agents, and to treat mountain sickness. 3. Cyclooxygenases COX1 and COX2 catalyze the synthesis of prostaglandins, levuloplandins, thromboxane, and so are clinically important enzymes. COX inhibitors such as nonsteroidal anti-inflammatory drugs (NSAIDs) are employed to offer relief from pain and inflammation. 4. Digestive enzymes like amylase, pepsin, trypsin, lipase, nucleosidase, nuclease, peptidase, and maltase are a few enzymes important for proper cellular processes. If a gene encoding for a particular enzyme required in a cellular process is defective, it may result in disease. Hereditary diseases such as albinism, phenylketonuria, fructosuria, methemoglobinemia, galactosemia, and others are the outcome of deficiency of an enzyme. Tyrosinase encoded by gene tier catalysis is the rate-limiting steps of melanin production. Missense mutation of TER gene causes albinism, which is a chromosomal disorder characterized by decreased pigmentation of hair, skin, and eyes. Phenylalanine hydroxylase POC converts phenylalanine into tyrosine in the body. So, mutation in POC results in excess accumulation of phenylalanine, finally resulting in a disease condition known as phenylketonuria. Fructosuria results due to mutation in the gene encoding for fructokinase, which degrades fructose into fructose 1-phosphate in the liver. Deficiency in reduced non-cytochrome B5 reductase results in rise in hemoglobin, in which iron is in the ferric Fe3+ ion form, causing methemoglobinemia. Galactosemia is caused when galactose degradation is inhibited due to defective lactose 1-phosphate aryltransferase, GALT, and accumulation of excessive galactose in the blood becomes toxic. Other immobilized on gold and silver coated nanoparticle was shown to have an optimum temperature of 80 C. Free amylase has 60 C and were reusable. Similar to alpha amylase, immobilized bercolderic acacia lipase on phenol-modified ordered mesopore silica efficiently resolved pure 1-phenylethanol enantiomer. Such bioconversion by immobilized enzyme was quite faster than the free enzyme. Nanofibers like polysurface nanofibers, polystyrene foam-like, and hydrogen cellulose nanofibers, and nanotubes such as carbon nanotubes and peptide nanotubes are also used as support for enzyme mobilization. In silico strategy for enzyme engineering is an emerging approach to overcome the constraints of commercially available enzymes, which include high activity, high selectivity, a high stability in organic solvents, and broad substrate specificity. Programs such as Metal Search, Dezymer, Rosetta, and Orbit are used for de novo designing of enzymes. Application of enzyme facilitates the production of pure enantiomers through high chemical-based selection, stereoselectivity, and regioselectivity. See table 12.41. Increased demands of enzymes for industrial production are compelling to adopt new strategies for enzyme technology. Various approaches such as mutagenesis, metagenomics, and computational biology have been useful in the bioengineering of enzymes for chemical processes. Of enantiopyrrhexadiol and atorvastatin, respectively. Two enzymes beta-aminase and beta-minosidase, displayed on the surface of yeast string, were able to hydrolyze 1,4-beta-D-mannan to produce ethanol. Biocatalysis efficiency for industrial applications can be improved by immobilizing enzymes to support. Immobilization can be done by any of the following methods. Figure 12.69. 1. Adsorption is the way in which the enzyme is attached to an inert material like glass and alginate beads. The concern with adsorption is that the active site of enzyme may be obstructed, reducing enzyme activity. 2. Enzymes can be immobilized by inducing the covalent bonding between the desired enzyme and matrix. This method of immobilization is the most efficient, which amplifies the enzyme activity. 3. Another technique of immobilization is entrapment, where the enzymes are trapped in insoluble beads or sol gels via hydrogen bonds, van der Waals forces, and ionic bonds. 4. Cross-linking technique of enzymes, namely cross-linking enzyme crystals, CELEC, and cross-linking enzyme aggregates, CLIA, is one of the approach for enzyme immobilization. Nanoparticles are also used to immobilize enzymes such as encoded magnetic nanoparticle. For example, alpha amylase when improvement method. Enzyme for industrial production examples. Directed evolution xylanase XYNA from B subtilis has been engineered through directed evolution for high thermal stability and pH tolerance. Immobilization lipase, a novel solvent-tolerant lipase from pseudozyme SPS, was immobilized on magnetic nanoparticles without loss in enzyme activity and prolonged shelf life. Protease produced by solid-state fermentation was immobilized on iron oxide nanoparticles. Metagenomic approach. Cellulase identification of novel cellulase genes from metagenomic library. Beta-glucosidase identified novel bromodolant beta-glucosidase from metagenomic library. Site-directed mutagenesis alpha amylase from B subtilis was improved for pH stability by mutation in histidine residue. Beta-glucosidase improved the glucose sensitivity and stability of trichodermal beta-glucosidase. Molecular modeling cellulase developed stochastic cellulase automata-based modeling approach to describe degradation of cellulosic material by cellulase system at single molecule resolution. De novo designed carbonic anhydrase. De novo engineered disulfide bonding bacterial type carbonic anhydrase for carbon sequestration. Microbial cell surface display manase. Cheetosanase MAMBI, aminase from Bacillus licheniformis DSM13, and CSNA, a cheetosanase from Bacillus subtilis ATCC 23857 were fused to different anchoring motifs of L-plantarum for covalent attachment to the cell surface for the development of whole cell biocatalyst. Fusion enzymes internal fusion of a yeast homolog of sumoprotein SMT3 could confer elevated optimal temperature and improved operational stability too. Desucrose 3 epimerase. Table 12.41 different improvement methods of enzymes for chemical production. Figure 12.69 methods of immobilization of enzymes. Enzymes are immobilized on a support or carrier by physicals such as van der Waals and hydrophobic interactions, ionic or covalent bonding. The supports generally used for immobilization are biopolymers, polysaccharide, inorganic solids, silica, or synthetic resins. Entrapment of enzymes is typically done with inorganic or organic polymer matrix like silica sol gels and polysaccharide respectively or microcapsule or membrane device. Cross-linking of enzymes aggregates or crystals, CLIA or CELEC, uses diffunctional cross-linkers. CLEAs or CLEX type of enzyme immobilization are highly stable and cost-effective as they do not involve the use of carrier. Enzymes for environmental waste and waste management. The concept of using enzymes for waste treatment has been in practice since 1970, though it was proposed 4 decades earlier. The reduction of pollution indicators, which includes biochemical oxygen demand, carbon oxygen demand, or total organic compound (TOC), is the measurement of waste treatment. The different types of environmental waste include solid waste, liquid sewage waste, and gaseous waste. Physical and chemical waste treatment processes can remove waste but at a low level, are costly and often used as polishing step. Due to high specificity, enzymes can selectively remove target pollutants. Prior to enzymatic treatment of the waste treatment, specific toxic chemicals should be removed to reduce disposal cost. The green method of effluent treatment is efficient in removing recalcitrant pollutants. The physiochemical treatment, including various steps of precipitation, coagulation, flocculation, and membrane filtration, is rapid, efficient, and controllable process, but is not economical. Since physiochemical treatment involves high energy consumption, increased soil waste, strict permissible limit of pollutants led to the development and utilization of enzyme-based waste management. Factors of environmental waste. Various industries like petroleum, chemical, textile, coal, paper, and pulp industries contribute to the accumulation of pollutants such as phenols, aromatic amines, and other aromatic compounds. For example, in pulp industry, the dark brown color of the pulp is removed by bleaching. This bleaching process results in the release of the toxic dark-colored effluents. Such effluents are hazardous to the environment, and enzymatic treatment eliminates such toxins, including the use of lactase and peroxidase. Waste from pesticide industries and cyanide release from the chemical industry, pharmaceutical, rubber, etc., are treated by parathion hydrolase, cyanidase, and cyanide hydratase. Solid waste, municipal sludge sewage contain contaminants like arsenic, copper, cadmium, lead, and chromium are removed by lipase, lysozyme, and phosphatase. Oxidoreductase, which oxidize by transferring oxygen electrons using fat, NADH, NADPH as substrate. They metabolize organic compounds, mediating increased solubility in water, reactivity, and also cleave the aromatic rings and dehalogenation. Monooxygenase is catalyst the oxidation of substrate by transferring electrons to complex molecules like fats and steroids. Monooxygenase catalyzes various metabolic processes such as dehalogenation, desulfurization, denitrification, ammonification, hydroxylation, and biodegradation of various aromatic and aliphatic compounds. Peroxidase is catalyst the breakdown of hydrogen peroxide, thus protecting the cells. Other enzymes used in waste management are lipase, protease, hydratase, lactase, cellulose, and esterase. Implementation of recombinant DNA technology for bulk production of enzymes would increase the efficiency of enzymes in waste management. As immobilization of an enzyme increases its reusability, so further research in this area of developing immobilization support would also aid in enzyme-mediated waste treatment. Microorganisms produce a variety of enzymes useful in effluent treatment, so to increase the production of such enzymes, microbes can be genetically modified. Methods of enzyme treatment. Immobilization of enzymes reduces the loss of enzyme and increases the reusability. Hence, waste management includes the utilization of immobilized enzymes. Several supports like gamma aluminium oxide pellets and magnetic beads are used to immobilize enzymes like lactase and peroxidase. Whole cell is also employed to treat effluents and is one of the easiest and simplest methods of enzyme-mediated waste management. Combination of enzyme technology and nanotechnology known as single enzyme nanotechnology (SEN) is also used for waste management.